Okay, welcome. Let's see the second very important approach or method that is used in drug discovery. I said for you that there are two very important methods or approaches that are used in drug discovery, mainly molecular modification. In molecular modification or association, the first one is general process. That includes molecular association or conjunction and molecular simplification or disjunction. The second major one is spatial process. Under this spatial process, there are two very important processes we have. The first one is alteration, which will increase or decrease the dimension and the flexibility of the molecules. The second one is other possible ways that are used to discover new compounds. Under this in special process, one way or one method, the first method that is used to discover or develop or manipulate new drug is ring closure or ring opening. What does it mean? We have a lot of examples of drugs that are discovered by either ring closure or ring opening. We have examples here. For example, here we have diphenhydramine. This is the drug that is recommended for treatment of motion sickness, nausea, and vomiting. This compound, it has this structure of diphenhydramine. When the diphenhydramine side chain forms a new compound, the side chain, the branch, it produces a new compound when it is forms a new ring by using the side chain. That is nephopam. This is a new compound. Therefore, diphenhydramine, when side chain it forms, or if the ring is closed, the side chain, by using the side chain, it will produce a new compound with a new maybe pharmacological activity, with the same pharmacological activity with better increase efficacy, increase stability. Therefore, the second very important thing is spatial process, as I said. Ring closure may produce a new compound with a new pharmacological activity, with a new desirable activity. Another one is ring opening. You can see here, I have also another example for this one. That is estradiol. Estradiol is this compound. When its ring is open, it produces diethyl ethyl estradiol. They have the same pharmacological activity. They are used as contraceptives. But in the case of estradiol, the rings, they are not open. But when the second and third ring, they undergo opening, they produce a new compound, which is called diethylstilbestrol. That is synthetic derivative of estrogen receptor agonist. Therefore, here the objective is one and a very important way in drug discovery is using either ring closure, closing the ring if you have side chain, or if you have many rings, therefore opening those rings. During that time, the new compound that is formed by opening of those rings, it may have activity. This is the first method in spatial process where we are discovering a new drug. The second one is formation of lower or higher homologous series. In another word, using homologous. Homologous are compounds, drugs, substances that differ from each other by the consecutive ones, by CH2 groups in the same families. Therefore, increasing the homologous or decreasing the homologous, it may have different pharmacological activity. It may produce different biological activity. Therefore, increasing the side chain by using CH2 group, or by decreasing this one, we can generate a new compound. This is the second method for drug discovery by special process. For example, this is the structure for alkyl trimethyl ammonium. This structure. This is when N is 5, it is muscarinic full agonist. I mentioned this. Agonists full. Agonists are agonists or drugs that can produce maximum response, maximum efficacy. Partial agonists are drugs that will activate the receptor, but they will not produce maximum response. Therefore, this compound, when N, do you see that here there is N? When N is 5, it is full agonist for muscarinic receptors. But when you change the value of N from 5 to 7 or 8 or 6, this activity will be changed. That means, I said, if N is 5, it is muscarinic agonist. When N is more than 5, that means 6 or 8 or 7, it will act as partial agonist. When N is 5, it is full agonist. When N is 6, 7, it is, or 8, partial. The agonistic activity, the activating power will decrease. Therefore, the agonistic activity is decreasing. But when N is 9 and above, the agonistic activity will lost. The drug will act as muscarinic antagonist. It will block muscarinic receptor agonists. It will block the receptor. It will not activate the receptor. Therefore, changing the homologous series, changing the chain, will change the pharmacological activity. This is one very important thing. Therefore, the other very important thing is, in a special process, is introduction of double bonds. Therefore, introducing or adding or removing the double bond will change the pharmacological or biological activity. Therefore, if you have a drug, if you add a double bond, the physicochemical property, the pharmacological activity may be changed. Or there may be a new activity. Here, whenever we introduce a double bond in a compound, there may be the probability of existence of geometrical isomerism with different activity. You know that cis and trans compounds, E or the Z compounds, they may have also different pharmacological activity. One is active, the other may not be active. Therefore, this is one possibility. Introducing double bond will produce cis or trans isomer. One may be active because of its configuration or conformation, the other may not be active. Therefore, we can take the active one. Therefore, by introducing the double bond, we can discover a new compound. The other very important thing is that you can see here this example, introduction of double bond. For example, I have here the example of diethylstilbestrol. These are the cis and trans isomers of the diethylstilbestrol. One is E, that means trans. The other one is Z, that is cis form. These compounds, they have the same molecules, the same number of atoms, but they differ on their orientation, their structural arrangements. Here, E is active, D is inactive. The cis is inactive. The trans is active, potent. It has activity because of orientation. Therefore, introduction of a double bond produces new activity, better activity. Here, why that the trans one is active and the cis one is inactive in the case of diethylstilbestrol? Because in drug design, we state that drug design means producing or making a drug based on its target site, we said. Therefore, when we see the structure of the E or trans diethylstilbestrol, naturally the trans structure resembles estrogen receptor. It is similar, look likes estrogen receptor. Therefore, it better fits the estrogen receptor. But because of the structural arrangement or configuration or conformation of the cis, it will not bind with the receptor. Because structure is not look like the receptor. It will not bind. Therefore, if it will not bind, it doesn't have activity. It will not produce effects. This is the second one. Therefore, introducing double bond on drugs, compounds, will produce a new pharmacological or biological activity. Therefore, by introducing a double bond, we can generate a new compound. That is another possibility, another way, another approach for discovering a new drug. Another one is when we introduce a double bond, there is also the probability of changing the physicochemical properties. Agonists, they may be converted into antagonists. Here we have an example, morphine. This is a structure of morphine, mu receptor agonist, nalorphine, antagonist of morphine. Therefore, introduction of a double bond here, there is a double bond here. Introduction of this double bond here will changes the pharmacological activity from agonist into antagonist. Therefore, introduction of double bond, it may have these two probabilities. Formation of cis-trans isomerism or geometrical isomerism. These geometrical isomers, they may have different activities. The second one is introduction of double bond may also produces or changes the physicochemical properties, agonist into antagonist. Also, introduction of chiral center, producing a compound to be chiral. Receptors are chiral entities, and the interaction of many drugs at the specific site exhibit chirality of interaction. Because of this one, clearly producing or synthesizing compounds that contain chiral center is also one very important way of discovering a new drug. This is also another very important procedure in drug discovery. The other one, introduction or removal or replacement of bulky groups. Some compounds, some drugs, they may have bulky groups. As we saw in the case of cocaine, these bulky groups, they may be responsible for the side effects. They may be responsible for short duration of action. They may be responsible for toxicities of these drugs. Therefore, removal or replacement of these bulky groups with suitable groups is also another way for discovering a new drug. Or introducing bulky group also. When you introduce a bulky group on a simple compound, in a simple drug, the activity may be changed. You can get also new compound with new pharmacological activity. This approach is used mainly to convert agonists into antagonists or antagonists into agonists. For example, I have two examples here. Tolazolin, alpha-adrenergic antagonist, and nafazolin, alpha-adrenergic agonist. You can see here the structure. This is tolazolin. This is nafazolin. When there is a bulky group introduction here, when we introduce bulky group here on tolazolin, it is converted into nafazolin with another pharmacological activity. Therefore, introduction or removal. When you remove this bulky group, you can get this compound. Introduction or removal of bulky groups may produce new compounds, new drugs. This is also the approach that is used for discovering a new drug. The other one, in the case of methylhistamine and cimetidine, cimetidine is histamine H2 blocker drug that is used for treatment of ulcer. Look here, this is the two structure. Here, introduction of this bulky group. This is a bulky group that is introduced here. Therefore, converts histamine receptor agonist into antagonist. Therefore, to produce agonists, to produce antagonists, so vice versa. We can use introduction, removal of bulky groups. When you introduce a bulky group, this may be changed into agonist or antagonist. When you introduce a bulky group, the agonist may be converted into antagonist. Therefore, these are some of the approaches that are used in drug discovery in medicinal chemistry. Other possible modifications that are used in special process in drug discovery, they are the following. Isosteric replacement. What are isoesters? Isoesters or bioisoesters are molecules or group of atoms that have the same number and arrangement of electron. Nitrogen with carbon monoxide. They have the same number of electron and the same arrangement. On nitrogen, N2 molecule, there are triple bonds. Three bonds that are bonded ones, electrons, triple bond. Also, carbon monoxide has a triple bond. There are two lone pairs on nitrogen molecule. There are also two lone pairs on carbon monoxide molecule. Those compounds or atoms or group of atoms that contain the same number and arrangement of electron, they are called bioisoesters. Therefore, in medicinal chemistry, introduction of bioisosteric group will produce another compound or will generate a new compound, a new drug. We have an example. In the case of aspirin, we will see aspirin and diclofenac, or salicylic acid and antralic acid. They differ on OH group at ortho position. OH and NH2, they are bioisoesteric, meaning they have the same number arrangement. Sometimes also we can use those groups that have the same size also. Therefore, replacing these bioisoesters will produce new compounds, new compound. This is also one very important method for drug discovery. Changing position or orientation of some groups. For example, here I have hydroxybenzoic acid. The first one is para-hydroxybenzoic acid. The first one, this one is called para-hydroxybenzoic acid. This is called meta-hydroxybenzoic acid. This is called ortho-hydroxybenzoic acid. These compounds, they differ on the position of OH group. The first one is at fourth position. The second one is at the third position. The third one is at the second ortho position. Therefore, changing orientation of some important groups on compounds may change the pharmacological activity, biological activity. These are mainly, there is a term which is called pharmacophore. Pharmacophore is the group which is responsible for pharmacological activity or biological activity. Pharmacophore is a group which is responsible for pharmacological activity or biological activity in a compound. In a drug, the group which is responsible for those things is called pharmacophore. Therefore, changing the orientation, changing the position of some important groups, some important side chains may change the activity, may change the pharmacological property, chemical property. Therefore, here the para and the meta, they are inactive, but ortho is active, which is called salicylic acid, the oldest analgesic drug that is commonly known. Therefore, here, this compound is active. Why? Because the OH group at the ortho position and the carboxylic carbonyl group, they can form intrahydrogen bonding that is useful for salt formation, that is useful for chelation. Due to this, when this compound, the ortho position is more potent. Intrahydrogen bonding means the hydrogen bonding that is formed in the same molecule. Interhydrogen bonding means if the hydrogen bonding is formed between two different compounds. In the same compound, we can say intrahydrogen bonding. Why the ortho is more active than the others? Because it forms intrahydrogen bonding that is important for formation of chelate, reactive molecule, reactive intermediate. This is also another way for discovering a new drug. The other one, modification towards inhibition or promotion of some various electronic status, changing the electron density, changing the affinity of some groups. For example, I have a drug M here. This is drug M. It has hydrogen. Replacing this hydrogen with more reactive electronegative elements like fluorine, chlorine, oxygen, nitrogen, sulfur. This may increase electron density on the drug. This electron density is very important for reactivity, interaction with the receptor, because they have lone pair of electron. Therefore, introducing elements that contain lone pair of electrons will increase interaction. This may increase potency, stability, duration of action. Therefore, mainly if you have a drug that contains hydrogen, replacing that hydrogen with the most electronegative elements will produce a new compound with a new substance. Uracil and fluorouracil. Uracil, introducing the hydrogen of uracil by fluorine produces antagonist. Therefore, this is the approach. Therefore, this is the approach for drug discovery and finding. Also, a very important thing is structure-activity relationship. What is structure-activity relationship? Structure-activity relationship, basically, activity of a drug either increase or decrease as a result of some structural changes relative to the prototype. Changing the structure may change the activity. This change is known as structure-activity relationship. That's very important in medicinal chemistry. And another topic is prodrugs. Introduction to prodrugs. What are prodrugs? Prodrugs are compounds that are biologically inactive But when they are metabolized, they will produce active substances, active compounds. They are classified into two broad classes: bioprecursors and carrier prodrugs. As I said, prodrugs are not drugs, but they are prior being a drug. Therefore, they are compounds, biologically inactive substances, but when they are metabolized, they produce active metabolite called drugs. The two broad classification of these prodrugs are the following: bioprecursor prodrugs and the carrier prodrugs. What is the difference between the two? What are carrier prodrugs? What are bioprecursor prodrugs? Bioprecursor prodrugs are compounds, prodrugs that already contain the embryo of the active species in their structure. If the compound contains in nature the active species in its structure, we can say that compound is a prodrug, but it is called bioprecursor. Compounds in nature, in their structure, if they contain the active species which can be metabolized after absorption or after administration, they are called bioprecursors. But carrier prodrugs are prodrugs that are formed, synthesized in the laboratory. For example, aspirin. It is formed by reacting salicylic acid with acetic acid. Salicylic acid is the active drug, but acetic acid is the carrier. Therefore, carrier prodrugs are prodrugs that are synthesized in the laboratory by combining the active species with the carrier to bring better pharmacological activity. Look here, example of bioprecursor. Cyclophosphamide. This is the most popular type of anti-cancer drug. It is an active compound, a prodrug. But this compound, cyclophosphamide, it contains the active species in structure. This is called phosphoramide, mustard, the active one. This compound in nature, it contains the active species. Sorry, this is the active species part. This is the active species part. This is the active species part. Therefore, drugs that contain naturally the active species in their structure, they are called bioprecursors. Carrier prodrugs, they are prodrugs that are formed by combining an active drug with a carrier species to form a compound with a desired chemical and biological characteristics. For example, to increase lipid solubility, to increase safety, to improve transport through membrane. We can combine the active species with the carrier. Here we have an example. Carrier plus active species will produce carrier product. This carrier product, after administration, it can undergo metabolism to furnish the carrier and the active species. The active species will act on the site of action, but the carrier can be eliminated or excreted. It must be non-toxic. When we are selecting the carrier, always you have to be careful. That carrier must be non-toxic. That can be easily eliminated from the body. This is the difference between carrier prodrugs and bioprecursors. Based on the bond, the linking, the carrier prodrugs, they are classified into two: bipartite prodrugs and tripartite prodrugs, or bipartite carrier prodrugs and tripartite carrier prodrugs. Bipartite prodrugs, they are prodrugs that are formed by linking the carrier with the active space by functional groups like ester, amides, simple functional groups. If the linking between the carrier and the active drug takes place by functional groups, by removing water, by removing some groups, we can say it is bipartite carrier prodrug. But if the active drug and the carrier, if they are combined with each other by the structure rather than the functional group, bulky structure, we can say it is tripartite prodrug. Bulky structure, not simple functional groups like ester and amides, but the two active drug and the carrier, if they are connected with bulky structure, another structure that connects the two, we can say it is tripartite prodrug. That is the difference between the two. Let's go. Why we can use prodrugs? Why cannot we can use? Why we can use the active drug as itself? What is the objective of prodrugs? What is the advantage of prodrugs? Why we cannot use the active drug as it is? For example, salicylic acid is the most common type of, or the oldest type of analgesic drug. Why we cannot use salicylic acid? Most of the time we can use aspirin. Salicylic acid is not used as a drug because it has side effects. It will produce gastric irritation and bleeding when the active drug salicylic acid is used. To overcome this problem, it is used in the form of prodrug called aspirin, because aspirin, it has very, very less side effect than the parent salicylic acid. It can pass the gastric wall and it doesn't have this gastric irritation and the bleeding. It's less side effect. And another advantage of aspirin over salicylic acid is absorption. It is better absorbed than salicylic acid. Let's see the objectives of, this is an example. Let's see the objectives of the use of prodrugs. The first one, the objective or the application, why we can use prodrugs. The first one is to improve absorption and transport through membrane. Therefore, to increase the transport. For example, we have lipid-soluble drugs, also hydrophilic drugs. Therefore, to increase lipophilicity of a given drug, we can combine our active drug with lipid-soluble carrier to allow the transport better. Therefore, if your drug is highly hydrophilic, for example, it will not pass to the central nervous system. There is a barrier called blood-brain barrier. If it is highly hydrophilic, therefore it will not pass to the blood-brain barrier. Therefore, to allow this drug to pass the blood-brain barrier, you have to incorporate your active drug that is hydrophilic with the carrier lipophilic. You can take the carrier lipophilic, or you can design lipophilic carrier. Whenever you combine the two, the lipid solubility will increase and it will pass the blood-brain barrier. This is one of the advantages of carrier prodrugs, or the advantages of prodrugs, to improve absorption, to increase transport. We can use prodrugs. The second one, the advantage or the objective of using prodrug is to improve patient acceptance. There are some drugs that are highly bitter. Also, when they are given by injection, they may produce irritation. Mainly those drugs that are highly bitter, they are rejected by children most of the time. Therefore, to increase patient acceptance, we can react the bitter drug with sweet substance like sugar. Therefore, your active drug is bitter, the carrier is sugar or sweetie. During that time, the test will be changed. Therefore, odor and taste are important aspects of drug administration. Therefore, increasing the taste, increasing the odor is another objective of prodrugs to improve the acceptance by patients is another very important thing. Therefore, look here, for this example, I have palmitic acid. This is a long chain fatty acid. Here, this compound, this palmitic acid, it has this structure. This is the active moiety. This is the active moiety. This is the carrier. This is highly bitter substance. Therefore, to increase its taste, it is incorporated with the carrier. This is active, this is a carrier by using ester bond linkage, by esterification. This is sugar, this is active drug. The sugar will act as a carrier. Therefore, the second very important objective in drug design is, or prodrug design is, to increase patient acceptance. The third one, to increase the duration of action or to slow release transient time. This is another very important objective of prodrug design, to increase the duration of action. We have a lot of drugs which are designed by this method. For example, look here. I have one example, that is tolmethine. This is tolmethine. Tolmethine sodium, or it's called tolmethine sodium. This compound, it has, this duration of action is 1 hour when it is given in the form of active drug. But when it is incorporated, or when it is given in the form of a prodrug, its duration of action is 9 hours. Here, the time, the duration of action is increased from 1 to 9 when it is given in the form of prodrug. When this is active drug part, this is active drug part, this is the carrier part, this is glycine, which is called tolmethine glycine prodrug. Therefore, the use of prodrug will increase the duration of action. Therefore, when the active drug is combined with the carrier, the duration of action may be increased. Therefore, this is the third objective of prodrug design. The fourth one, to increase specificity, site specificity, target specificity. In these theories, it should be possible to design a carrier product that would only release on the site of action, the drug in the vicinity of its site of action, to allow a drug to reach to a specific site. We can use prodrug approaches. Another and the last one that is very important thing in prodrug design is to minimize the side effects or to decrease toxicities of drugs. We can use prodrugs to minimize side effects. Therefore, look here, this is the structure of salicylic acid. This is the oldest analgesic drug. The oldest analgesic drug. This compound, when it is administered, it will produce gastric irritation in patients and bleeding. To overcome this side effect, they changed this compound into prodrug, which is called aspirin. Therefore, conversion of salicylic acid to its ester called aspirin reduces the degree of stomach irritation and bleeding. and also increases absorption. Therefore, here, a very important thing is whenever a drug is, mainly if it has side effects or if they have toxic nature, converting that active drug into a prodrug will minimize these unwanted side effects. And why we can always in medicinal chemistry, most of the time we will talk about new drugs. Here also I'm talking about new drugs, new drug. Why is that? We have a lot of drugs. For example, we have a lot of antibacterial drugs. We have a lot of antiviral drugs. We have a lot of anti-malarial, anti-cancer. We have many antifungal drugs. Why we need new drugs? What is the objective of searching for a new drug? The first one is to combat, to overcome resistance. We can search, we need new drugs. The objective is to overcome, to combat the resistance. The second one is to improve treatment for existing diseases, to improve the activity of drug, to improve stability, to improve efficacy, to improve duration of action of drugs for treatment of the existing. We need a new drug. Also, the other objective of why we need new drug is for treatment of newly identified diseases. We need new drugs for Ebola, for Zika virus, for coronavirus. Therefore, these are new diseases. If there is a new disease, we can search for a new drug that is used for treatment of this. Also, the other, to produce safer drugs. Because we have drugs that have a lot of side effects. Therefore, to overcome this overdose toxicity or side effects or adverse side effects of drugs or existing drugs, we need new better drugs that can substitute these existing drugs. This is the reason why we need new drugs. In drug discovery and development processes, there are different stages, phases. These are called drug discovery and development phases. The first one is in drug discovery and development. The first stage is called target identification. The second one is lead compound identification. The third one is lead optimization or lead modification, or you can say lead development. And after passing this lead optimization, we can pass into another stage which is called preclinical trial on animal models. The fifth one is clinical trials. Then after clinical trials, we have nomenclature, registration, manufacturing, marketing. These are the common stages, mainly in medicinal chemistry and other books. I saw that there are 5 major things, major phases in drug discovery and development. These are, the first one is target identification, lead compound identification or selection, lead optimization, preclinical trials and clinical trials. After that, we have also another phases, nomenclature phase, registration phase, and manufacturing. There are about 13 phases that are commonly used in drug discovery and development. Let's see each of these. The first phase in drug discovery and development is called identification of biological target. What does it mean? Drug target is a specific macromolecule or biological system which the drug will interact with proteins, genes, channels, receptors. It means the place where the drug will act. The place may be DNA, maybe RNA, maybe enzyme or ion channel in which the drug can undergo interaction. This is called biological target. Therefore, the first thing in drug discovery and development is identifying the target. Where we can use this drug? In which place? Where is the target? Is the first stage in drug discovery and development. Identifying the target organ, the target site. After identifying the target site or the target's biological target, the second one is lead compound identification. Which one is the lead compound? The lead compound is one that has basic structural requirements for exhibiting the desired action against the chosen target. The structure that can have activity on that selected identified biological target. Identifying or selecting the lead compound. Which one is the lead compound for that target? This is called the second stage, lead compound identification. This is the second, very important. Then, after this, lead compound modification or lead compound manipulation, the third stage. Therefore, here in this lead structure optimization or lead structure modification, if the compound, if it has activity on the target site, it may have side effects or its activity may be lower. To enhance its activity, to enhance its stability, to enhance its efficacy, to decrease the side effect, we have to modify, we have to optimize, to increase desirable pharmacological activity and decrease undesirable, unwanted side effect from that molecule. This is called lead structure modification or lead structure optimization. And the fourth one, preclinical test. Testing the new drug, the new compound in animal model. This yields information on the biological activity of a new substance or new compound. Therefore, testing the activity, the potency. Also, to explore the drug's efficacy and safety. The new drug's efficacy and safety before using in human. We can use this test, preclinical test in animal, to check its efficacy, safety. And this takes place in vitro, in animals, to check the pharmacodynamic properties, pharmacokinetic properties, toxicologies. These takes place in preclinical test. The toxicology is that the compound is toxic. If it is toxic in animal model, we cannot use in human. We cannot pass into the clinical stage. Therefore, we have to go the first modification stage. Therefore, molecular modification also again will be takes place. Then IND approved by Food and Drug Administration to proceed to clinical trial. If that compound in preclinical stage, if it is safer, if it is better, if it has better things, after seeing the pharmacodynamic, pharmacokinetics, toxicology, if it has better activities, desirable activity, meaning that if it has some better activities, we can pass to the other stage, which is called clinical stage. That means, which is called, if the drug it has positive thing in preclinical stage, it is called new drug, investigational new drug, IND, investigational new drug. This investigational new drug is approved by Food and Drug Administration to pass to the another stage, clinical trial. Under this, testing a new compound in clinical trials on human. There are 4 phases: phase 1, phase 2, phase 3, and phase 4. The first phase is called phase 1. In this case, the new drug is taken, or the new drug was tried on healthy volunteers. The activity, is that activity similar with the animal model? To determine whether effect observed in animal experiment also occur in human, to establish the dose level at which signs of toxicity first appear, to investigate metabolism, pharmacokinetics, biological effects. We can use this new drug in the activity of this new drug in phase 1, mainly in healthy volunteers, to determine its effect, whether it is similar with animal model. Phase 2, the new drug is administered to patients for the first time. Then dose response studies takes place in this phase to check potency efficacies. Also, adverse effects, they will be managed here. Monitoring subjects for adverse effects takes place in phase 2. Phase 3, the number of patients will increase here, maybe hundreds and thousands patients. The new drug will be compared with standard drugs to compare the activity, the stability, the potency of new drug with the standard drugs, to verify the efficacy of the drug and to detect effects that may not have surfaced in the phase 1 and the phase 2 trials, during which exposure to the drug was limited. New investigational drug to drug regulatory bodies will be submitted. Then, if it has similar effects or there is maybe also some differences, therefore it is approved by Food and Drug Administration, takes place in phase 3. Then for serial manufacturing also will take place sometimes in this stage. And the 4th stage, post-marketing. Post-market surveillance, checking the side effects, problems that are associated with drug-drug interaction and other toxicities. They are studied under this post-market surveillance, observing long-term effects of the drug on the general population after approval. And if there is side effects, the professionals, they will report. If there is side effect also, no side effects, they will report for Food and Drug Administration. Then renewal and cancellation of the license of a drug. If it will be renewed if it has desirable pharmacological activity, and cancellation if it's not proper for use.